A lysosome, derived from the Greek words lysis, meaning digestive, and soma, that is body, is a membrane bound cell organelle found in most animal cells. They are absent in red blood cells. Structurally and chemically, they are spherical vesicles containing hydrolytic enzymes capable of breaking down a wide variety of particulate materials, including proteins, nucleic acids, carbohydrates, lipids, and cellular debris, and thus perform intracellular digestion of the cell. They are known to contain more than 50 different enzymes, which are all optimally active at an acidic environment of about pH 4.5. Thus, lysosomes act as a waste disposal system of the cell by digesting unwanted materials in the cytoplasm, both from outside of the cell and obsolete components inside the cell. Thus, lysosomes are the recycling units of a cell. For this function, they are popularly referred to as suicide bags or suicide sacs of the cell. Furthermore, lysosomes are responsible for cellular homeostasis for their involvements in secretion, plasma membrane repair, cell signaling, and energy metabolism, which are related to health and diseases. Depending on their functional activity, their size can be very different. The biggest ones can be more than 10 times bigger than the smallest ones. They were discovered and named by Belgian biologist Christian de Duve in 1955, who eventually received the Nobel Prize in Physiology or Medicine in 1974. Enzymes of the lysosomes are synthesized in the rough endoplasmic reticulum. The enzymes are released from Golgi apparatus in small vesicles, which ultimately fuse with acidic vesicles called endosomes, thus becoming full lysosomes. Acrosome of sperm cell is also a modified type of lysosome. Occurrence: usually the lysosomes occur in most animals except the mature mammalian red blood cells. The animal cells which are secretory in function, such as the pancreatic cells, leukocytes, liver cells, spleen cells, and kidney cells, contain large number of lysosomes. These are particularly numerous and large in cells as macrophages, which perform a special digestive functions. The lysosomes often remain distributed evenly in the cytoplasm. Morphology, shape and size. The lysosomes are generally spherical in shape, but certain meristematic cells of plant roots contain irregular shaped lysosomes. The size of the lysosomes usually ranges from 0.2 to 0.8 micrometer, but may be exceptionally large in the cells of mammalian kidney, that is upon 5 micrometer, and even more in phagocytes and leukocytes. Structure: the lysosomes are round vacuolar structure which remains filled with a dense material and acid phosphatase enzymes. The lysosomes are bounded by a unit membrane of the lipoprotein. The space or vacuole of the lysosomes contain dense, granular, vacuolated and homogeneous material. Chemical composition: the lysosomes have been found to contain 50 different enzymes or hydrolases. The one most often used as a marker because of the ease with which its activity can be localized in electron microscopy is acid phosphatase. Some important lysosomal hydrolases are beta galactosidase, beta and acetyl glucosaminidase, alpha glucosidase, alpha mannose, cathepsin A, cathepsin B, aryl sulfatase A, aryl sulfatase B, acid ribonuclease, acid deoxyribonuclease, acid phosphatase, acid lipase, phospholipase A, phosphatidic acid phosphatase, hyaluronidase, phosphoprotein phosphatase, aminopeptidase A, dextranase, saccharase, lysozyme, magnesium activated ATPase, indoxyl acetate, esterase and plasminogen activator. All these enzymes of the lysosomes are enclosed within the single lipoprotein membrane, which can be ruptured easily by progesterone, endotoxins, testosterones, etc. But the lysosome remains stable in living cells. The stability to the lysosome is provided by certain chemical substances such as cholesterol, cortisone, cortisol and chloroquine. These substances stabilize the membrane of the lysosomes. Let us now talk about the kinds of lysosomes. In different types of cells and at different times in the same cell, 4 types of lysosomes have been observed. They are primary lysosomes, secondary lysosomes, autophagic vacuoles and residual bodies. Primary lysosomes. Newly formed lysosomes are called primary lysosomes. They are formed principally as vesicles pinched off from Golgi membranes, but in some cells a specialized portion of endoplasmic reticulum called GERL, that is Golgi associated endoplasmic reticulum, from which lysosomes arise, is probably also involved. Secondary lysosomes. The secondary lysosomes are also known as heterophagosomes or digestive vacuoles. When the cells feed on a foreign or exogenous or extracellular substances by the process of phagocytosis or pinocytosis, then the plasma membrane forms the membrane bound vesicles known as pinosomes or phagosomes around these substances. The pinosomes or phagosomes ultimately fuse with the pre-existing primary lysosomes and form the secondary lysosomes or heterophagosomes. The objective of secondary lysosome formation is to mix lysosomal hydrolysis with the ingested material, so that breakdown of the latter can occur. The digestion products like amino acids, nucleotides has a low molecular weight and readily pass through the lysosomal membrane to become sources of energy or raw materials for biosynthetic pathway. As product leave, the lysosome often diminishes in size. This change requires a loss of surface area, which can be achieved by an inward budding of the limiting membrane in much the same way as pinocytic vesicles are formed. The internalized membranes is then digested. This process, which is particularly prominent in the digestion of the pinocytic vesicles, leads to an electron micrographic picture of a vacuole containing a numerous small vesicles and fragments, a structure known as multivesicular body. The fusion of a lysosome with an endocytic vacuole is a selective process. The vacuoles do not fuse with endoplasmic reticulum, mitochondria, etc., nor do the lysosome fuse with a plasma membrane under ordinary circumstances, even though plasma membrane and phagocytic vacuole are made of the same stuff. However, in the presence of cytochalasin B, extracellular release of lysosomal enzymes does occur readily, indicating that perhaps the microfilamentous web beneath the plasma membrane normally protects it from fusion with lysosomes. There is some evidence also for involvement of microtubules in guiding endocytic vacuoles, lysosomes and specific granules, and there is evidence for regulation by cyclic nucleotides, probably via their effect on microtubules. Killing in a secondary lysosomes. Simple enzymatic digestion by hydrolysis is not the only process that occurs in a secondary lysosomes. Many microorganisms, while they are viable, resist attack by hydrolysis. Hence, mechanisms have evolved specifically to kill ingested organisms prior to their breakdown. Lysosomes typically contain lysozyme, an enzyme that degrades bacterial cell wall, especially the simpler walls of gram-positive bacteria. The lysosomes of neutrophils also contain a lactoferrin, which binds iron and other metal ion tightly, thus inhibiting growth of the ingested organisms. The contents of the digestive vacuoles are usually very acidic, that is less than pH 4 in neutrophils, which by itself inhibits the growth of many bacteria. And finally, oxidases thought to be present at the surface of the cell membrane and thus also on the inside surface of digestive vacuoles produce hydrogen peroxide is microbicidal in its own right. But in neutrophils and most macrophages, it is also utilized by an enzyme called myeloperoxidase to covalently fix chloride or iodine onto organic targets such as bacteria. This halogenation greatly facilitates killing of the organism. Autophagic vacuoles. Lysosomes are involved not only in the process of digesting extracellular material known as heterophagy, but also in the digestion of intracellular substances and activity known as autophagy. In autophagy, lysosomes are responsible for turnover of organelles of animal cells, a process that is accelerated during involutional states in a muscle that is not being used, during starvation, after injury, or during the remodeling that occurs with cellular differentiation. Even in unstressed normal cells, the turnover of organelles by autophagy is relatively rapid. The half-life of a liver cell mitochondria, for example, is 5 to 6 days. For peroxisomes, it is 1 to 2 days, and for ribosome about 5 days. The autophagy of cytoplasmic organelles start with an isolation envelope, which is a sac of smooth surfaced membrane that wraps around the material to be removed and separates into a sort of phagocytic vacuole known as an isolation body. The source of the isolation membrane, at least in a damaged liver and kidney, seem to be endoplasmic reticulum. However, work of Michael Locke and his associates in 1975 indicates that the isolation membrane in an insect cell during metamorphosis and in some other animal cells is derived from the Golgi complex. In any case, lysosomes can fuse with the isolation body to create an autophagic digestive vacuole and release a breakdown products to the cytoplasm to be used as an emergency source of food or as a raw material for cellular remodeling. Residual bodies. Although killing and breakdown in the lysosomes are efficient, some materials are simply not digestible. These substances may be released from the cell by fusion of the digestive vacuole with the plasma membrane by exocytosis. This process is very efficient in protozoa, but not so efficient in higher animals. Hence, inactive digestive vacuoles containing debris may accumulate. These debris-filled organelles called residual bodies are of no consequence to cells like macrophages or neutrophils because the cells do not live long enough for significant amounts to accumulate. In cells that do not divide and are not replaced, substantial quantities of these materials may accumulate. In neurons and muscle cells especially, there is a steady increase in the number of these bodies, which are called in this context lipofuscin granules. The age of an animal can often be estimated from their concentration, and residual bodies play important role in the process of aging. Let us talk about origin of lysosomes. The origin of lysosomes is not known with any amount of certainty. Some workers have suggested that the lysosomes have originated from the pinocytic vacuoles called pinosomes, which in their turn originate from the plasma membrane. Others believe that because the acrosome of the sperm, which contain a high concentration of acid phosphatase, is formed by the secretory activity of the Golgi complex, the lysosomes have Golgi origin. Still others consider the lysosomes are inclusions containing digested material. It seems likely that the membrane part of the lysosome is derived from Golgi membranes and the enzymes part from secretory products of Golgi complex. Novikov in 1965 has shown that the lysosomes originated directly from the granular endoplasmic reticulum. Lysosomes and disease. Defects in a normal function of lysosomes often cause a variety of pathological conditions in the animals, including man. The importance of hydrogen peroxide in phagocytic killing of microorganisms is illustrated in humans by a X-linked recessive condition called a chronic granulomatous disease. A granuloma is a collection of dead and dying macrophages. The disease is a deficiency of the oxidase enzyme that produces hydrogen peroxide in the digestive vacuole. The macrophages and neutrophils of these individuals are unable to kill effectively even common bacteria, which may live and multiply inside the cell, eventually killing it instead. Such people are prone to all sorts of fungal and bacterial infections in spite of an immune system that is otherwise normal. Other types of defects in lysosomal function include a failure to fuse with the endocytic vacuole and failure to digest a certain classes of chemical substances due to lack of one or more critical enzymes. For example, fusion of lysosome and phagocytic vacuole is sometimes inhibited for reasons that are not understood. When the vacuole contains live bacteria responsible for tuberculosis or leprosy, or when it contains live toxoplasma, a common protozoan parasite contracted from raw meat or a cat feces. Lack of fusion permits these organisms to live within the phagocytic cells and often to destroy them and to establish active diseases. Algae that live symbiotically in protozoa seem to be similarly protected. The only clue how fusion might be avoided in these cases come from the observation that concanavalin A induced endocytosis also results in vacuoles that frequently fail to fuse with lysosomes. Concanavalin A or Con A is a lactine, the plant protein that behaves very much like antibodies in their ability to bind to specific chemical configurations such as alpha D mannose or alpha D glucose of a plasma membrane and thus cause a cellular adhesion in plant cells. Con A is found to cause activation of lymphocytes by cross-linking with receptors of lymphocyte plasma membrane. The latter can anchor to the underlying cytoskeleton. However, high dose of Con A immobilizes many receptors of lymphocyte and prevents activation. Fusion of phagocytic vacuoles with lysosomes and specific granules may also come too soon. If fusion occurs before the plasma membrane has closed over the forming vacuole, contents of the lysosomes and specific granules may regurgitate through the vacuole and into the extracellular space. Because of the destructive character of these enzymes, a local inflammation, that is redness, swelling, tenderness, etc. may result. This process, especially common with neutrophils, is important in localizing an infection and hence not always undesirable. However, it is probably also responsible for some of the degenerative joint changes in rheumatoid and gouty types of arthritis. The effectiveness of colchicines in the treatment of gout is probably due to its inhibition of lysosomes and granule movement and fusion, which are processes that involve microtubules. Another type of defect in lysosomal function occurs if an essential hydrolase is missing. Because there are many hydrolases, there are many, more than 24 of these lysosomal storage diseases. They are almost always inherited as autosomal recessive traits, because one good gene out of the 2 alleles inherited from the 2 parents is usually enough to prevent symptoms. The diseases have names like Tay-Sachs, Hunter's, Hurler's, Niemann-Pick, and so on, because they were described long before the lysosomal functions was understood. Most of the diseases result in accumulation of undigested lipids or polysaccharides, which eventually fill the cell and thus compromise its function. The types of cell most affected will be determined by which enzymes is faulty, but the central nervous system is commonly affected, resulting in a progressive lack of body control and loss of a mental function, leading to a slow death. Now, let us look at the function of lysosomes. The important functions of lysosomes are digestion of large extracellular particles. The lysosomes digest the food contents of the phagosomes or pinosomes. The lysosomes of leukocytes enable the latter to devour the foreign proteins, bacteria and viruses. Digestion of intracellular substances. During the starvation, the lysosomes digest the stored food contents, that is proteins, lipids and carbohydrates of the cytoplasm and supply to the cell necessary amount of energy. Autolysis. In certain pathological conditions, the lysosome starts to digest the various organelles of the cells, and this process is known as autolysis or cellular autophagy. In the process of metamorphosis of amphibians and tunicates, many embryonic tissues, for example, gills, fins, tail, etc., are digested by the lysosomes and utilized by the other cells. Extracellular digestion. The lysosomes of a certain cells, such as sperm, discharge their enzymes outside the cell during the process of fertilization. The lysosomal enzymes digest the limiting membranes of the ovum and form a penetration path in the ovum for the sperms. The chondrioblast cell and osteoblast cells digest the cartilage and bone respectively by the process of extracellular digestion. The lysosomes are supposed to initiate the mitosis in cells. The lysosomes form a built-in mechanism of the cell and enable the cell to adopt a metabolically rapidly changing conditions of food supply, oxygen concentration and other environmental factors which occur in many living systems. Synthesis of lysosomal enzymes is controlled by nuclear genes. Mutations in the genes for these enzymes are responsible for more than 30 different human genetic diseases, which are collectively known as lysosomal storage diseases. These diseases are due to deficiency in a single lysosomal enzyme that prevents a breakdown of a target molecules. Consequently, the undegraded materials accumulate within the lysosomes and often giving rise to a severe clinical symptoms. Further, such genetic defects are related to several neurodegenerative disorders, cancer, cardiovascular diseases and aging related diseases. Recycle the products of a biochemical reactions that have taken place following materials being brought into the cell by endocytosis. Different materials are processed in a different ways. For example, some structure may be processed or degraded within the lysosomes and others are taken to the surface of the cell. In summarize, it can be said that lysosomes are membrane enclosed organelles that contain an array of enzymes capable of breaking down all types of biological polymers, proteins, nucleic acids, carbohydrates and lipids. Lysosomes functions as the digestive system of the cell, serving both to degrade materials taken up from outside the cell and to digest obsolete components of the cell itself. In their simplest form, lysosomes are visualized as a dense spherical vacuoles, but they can display a considerable variation in size and shape as a result of differences in the materials that have been taken up for digestion. Lysosomes thus represents a morphologically diverse organelles defined by the common functions of degrading intracellular material. So, this is all about the organelle lysosomes. Thank you.